Um,
Yeah, you can go ahead.
I
Sorry.
can go ahead.
Yeah.
Good morning. My name is Tanya, and I'm Franco's mentor. So I've been working with Franco this last weeks that he's been with us at Steinhauser Lab. Not only me, but Franco has been interacting with all the lab members, and it was a pleasure to have Franco with us. So since the very first day when we were talking about the projects that we have at the lab, and we are a quite diverse lab, Franco was asking questions, and he was actually asking really smart questions. So it was very promising from the beginning. And then Franco just engaged in one of the projects that we have in the lab, which is studying. We are trying to investigate new regulators of bone loss. And one of the means that we do that is by fasting. So we study the effects of fasting on bone. And Franco just jumped into this project. It has been over a year project. So Franco just helped us with that by conducting an experiment. And this is the experiment that Franco will be presenting today. So I hope that Franco had fun with us. We had fun. He's always welcome to come back. So Franco, good luck, and the floor is yours.
Okay. So once again, my name is Franco Alvarez. I was in the Steinhauser lab this summer in Bridgeside Point 1 in the Aging Institute. My mentor is Dr. Tanya Moram, and my PI was Dr. Matthew Steinhauser. And this summer, my project was studying the effects of methionine on osteoblast differentiation. So like my mentor already kind of glossed over a little bit, all this started whenever we were researching fasting and the effects of fasting on the body. There's a lot of research showing promising benefits in terms of fasting to many areas of the body, and a couple chronic conditions such as obesity, diabetes, cancer, and hypertension. But one of the things that we were more focused on was the negative effects of fasting on bone health and how it can seem to cause a lot of bone loss. We want to look at how we can maybe reverse that or stimulate bone growth. After a 10-day fasting study, we discovered that P1NP, which is a bone formation marker, decreased in healthy patients. And inversely, we saw that the essential acid methionine actually increased, which was kind of our first clue to how we could possibly treat this bone loss phenomenon. So that was our goal this summer, was to just dive into that and study it, see what's going on. And we wanted to find out whether methionine would change how bone forms. We were studying positive or negative impacts for that matter. And just because I think everyone here is on a little bit different of a level, I'm just going to start off with the basics on bone. There are three major types of bone cells: osteoblasts, osteoclasts, and osteocytes. Osteoblasts are important to us because they are the bone forming cells. That is what we really wanted to study because we're interested in healing bone and increasing bone mass. Osteoclasts, although they sound intimidating, are still very important to bone health because if you don't have a balance of osteoblasts and osteoclasts working together, that's when you start having imbalances in bone. You can have bone loss, too much bone. And so you definitely need all the cells working together to keep bone healthy. And osteoblasts, like I mentioned, are the cells responsible for forming bone. This is the differentiation process. This summer, we studied just the effects of methionine on this process right here, from pre-osteoblasts to mature osteoblasts. This was about a 16-day process. And by the end, we had mature osteoblasts if the treatments worked well. Like I mentioned already, when osteoblasts malfunction or they don't work properly, you get that imbalance in bone, and that can lead to bone loss, which is why we are so interested in studying why osteoblasts stop working or how we can treat them so that they start working better. So overall, this summer, my aim in my study was to investigate the effects of methionine on osteoblast differentiation. And my hypothesis from the P1NP and methionine levels that we saw at the beginning was that excess methionine decreases osteoblast differentiation. So this summer, this was the exact setup I had. I had 2 6-well plates set up exactly like this. 4 different treatments: no methionine, 20 micromoles, 100 micromoles, and 500 micromoles. We plated the exact same amount of human pre-osteoblast cells. It's from this cell line that probably doesn't mean a whole lot, but if you're a science person, it does. We plated the same exact amount of cells in each well, And then we added 4 different treatments of methionine, and this was included in the media. So as we changed the media every 3 days, the cells had fresh food to kind of grow from. They differentiated in a 39.5 degrees Celsius incubator for 16 days, like I mentioned, until they were fully differentiated. And at the very end, we pulled the plates out, stained it with alizarin red, which stands for calcium deposits or mature osteoblasts, because it's all kind of tied together. And we just looked at which wells had the most alizarin red for the most amount of differentiated osteoblasts. On the final day, we took some microscope pictures. These are what our wells looked like. I forgot to include this for the people that were here last week with me, but this is pretty important to look at because you can actually see how no methionine really affected the differentiation process. And there's a lot less cells compared to the other 3 treatments. It's hard to see with the eye between the other 3 treatments and even with the next picture too with the staining. But the most important thing you can take away from the pictures, I think, is that no methionine heavily impacted the way that osteoblasts differentiate. And there was a lot less like cell proliferation, and there's just not many cells left by the end. So once we did the readings, we found that the most significant data was that pre-osteoblasts treated with no methionine were significantly less like proliferating by the end of the differentiation process. And excess doses of methionine did have less differentiation, but whether this data was completely significant, we would need to do more research on to figure out whether our hypothesis was completely correct or not. You can also see that 20 micromoles was the optimal level for our osteoblast differentiation. But the most significant thing to look at from this bar chart, I think, is that no methionine was the largest inhibitor in whether osteoblasts were able to differentiate successfully. So when it comes to the end, methionine clearly does have an impact on osteoblast differentiation. We saw that in our study, we needed an optimal level of about 20 microliters. And it does appear that osteoblasts do not differentiate well in the absence of methionine, which is different than our hypothesis, which we may need to do more research on to figure out whether that's true or not. Like I said, our hypothesis was never really like disproved or proved, but our lab is continuing to do more research on whether this effect is true in not only cells, but also biological systems like mice and other systems to see if what we see in the cells is true in the actual body. And yeah, that's all. We have time for questions.
I may be remembering wrong, but I think you call methionine an essential amino acid in order to.
Yeah.
What does that mean?
Well, essential amino acids are used to create proteins throughout our body. So whenever methionine is used to create proteins, those proteins could be used to help our bone, I think. I know methionine is like tied to the start codon for most proteins. So methionine is really important in making proteins, but in terms of our study alone, we were just looking at how methionine affects osteoblast differentiation. Yeah. Well, we're doing a current study on mice to see whether these effects are true within the actual like living, breathing organism. We're still working on that. Yeah, that would be really important in looking at what the optimal levels are for osteoblast differentiation. I only had 4 treatments just to keep it kind of simple. Now that we kind of saw our results and saw that 20 microliters was like the most optimal level, we might do more research to see, like, find the balance of exactly which levels are the most optimal. But that would require more research, obviously. Did you have a question? I'm sorry, I didn't hear you.
So I noticed that the conference was carried out at 39.5. What was the reason for doing
That starts the 39.5 degrees Celsius is the temperature that premature osteoblasts begin to differentiate. I want to pull up the slide just to point it out again. To start this process right here at the beginning, that's where the 39.5 degrees Celsius comes in, because that's what starts this whole process. That's where the cells are most happy to continue growing, these specific types of cells. The only thing I studied this summer was actual cell work. So as far as applications in the whole body, I'm not too familiar with.
Good job. All right, Sophia, you're up.
Yeah, there's some
very useful that
Yeah, the projectors will get you. Here.
Okay. So my name is Dorota Jaswinska. I work in the Surveillance Lab, and I had the pleasure of working with Sophia this summer. She got a little bit of an introduction to bioengineering, since that is what she wants to do whenever.
Apparently, this is the one before his. Okay, is that all right?
Yes, I'd like to see it.
Is somebody talking to us? And mute. Mute. Maybe that one. I don't know why we can't mute that person. Mute.
I guess. Sorry for that interruption. So Sophia got a little bit of an introduction to bioengineering and medicine because she wants to go to medical school later on. And we looked at ovarian cancer metastasis, since that is my thesis project, and she took a little bit of it and looked at how MMT works with ovarian cancer. So thank you.
I think that'll stay. So as Dorota already introduced, my name is Sophia Alvarez, and I was in the Zerva Lab this summer, which is in the Center for Biotechnology and Bioengineering building, and it's literally the one like 2 doors down. And again, this summer, I studied the effects of mesothelial to mesenchymal transition on high-grade serous ovarian cancer metastasis. Let me just press the next one. Okay, sorry. So just a real quick overview. Ovarian cancer. Every year annually, there are 21,000 women in the U.S. diagnosed with it and about 14,000 deaths on average. Ovarian cancer is the 8th most common type of cancer, but the 5th leading deadliest cancer. Often it's not diagnosed until advanced stages, which is what makes it so fatal because we can't catch it at early stages, so it's hard to treat and stop. The most common and deadliest type of ovarian cancer is high-grade serous ovarian carcinoma, which is the cancer that we were studying this summer. And high-grade serous is a type of epithelial cancer that originates in the fallopian tubes, which these are the fallopian tubes here. One of the ways that high-grade serous is able to metastasize is through the peritoneum, which just lines the abdomen, as you can see over here. This peritoneum is made up of a monolayer of mesothelial cells, which is one of the key points that we studied this summer. And as you can see here, if the primary tumor is here, it can break off and go through the mesothelial layer that makes up the peritoneum and metastasize to other parts, as you see here throughout the body. Another focus point that we had was MMT, which is just mesothelial to mesenchymal transition. And mesothelial cells, as I already mentioned, they often form monolayers in the body around vital organs. And mesenchymal cells are more stem-like and more motile, so they want to move around more. And if you've ever heard of like mesenchymal stem cells, they're usually the cells that differentiate into other types of cells in the body. And you can see here MMT shown, where here are the mesothelial cells, and you can see them transition into mesenchymal cells over here. One of the main ways that we were able to study this summer was through microfluidics, which is like the bioengineering part of it a little bit. And this is the top view of the devices that we had. They're very little, probably only about like this big. And some advantages of using a system like this is that it's shown in 3D rather than 2D, and it allows for cells to be placed in a certain way. So rather than in a plate, just kind of putting cells on top of each other, we can actually like place them in places such as these channels here to watch how they interact with each other, again, rather than just being kind of mixed together in a plate. So as you see here, the channels, this is where we put the mesothelial cells, and we can put cancer cells in there as well. And then this gel region has collagen in it, and you can watch the cells actually kind of move around and invade and just kind of see how they interact. And these posts here are just to kind of keep that gel in so it doesn't leak into the channels. And then again, why we used it is because it simulates an in vivo assay, and also can predict similar outcomes to an animal without actually having to use animals. And this is from a different paper that worked with breast cancer, just to show you the 3D component of it. You can see that here would be the channels where we put the cells, and this is the middle channel wherever we have the collagen in the cells, and whatever else is in there can move around, just to kind of give you a visual of that. So our hypothesis was that high-grade serous cancer tumor cells can transform mesothelial cells to mesenchymal cells, and that this interaction between the tumor cells and mesothelial cells allows for increased metastasis. Here's one of the first things we did, was just tissue staining. The lab actually had like in vivo experiments in the mice, and so they cut up the tissue and put it on slides, which I was able to stain and actually look at. So the first thing that we do is deparafinize the tissue, which is essentially kind of removing the wax, and you start it with xylene and move it to lower grades of ethanol, so that way the stain will actually go into the tissue. The next part is quenching, which Jordan explained to me is kind of like boiling the tissue, and it literally looks like a pressure cooker, and we have to write not for food on it because it looks like a pressure cooker. And then after that, we block the tissue. This is to block non-specific targets, so that way whenever we stain, we're not staining all the cells in the tissue, we're only staining for the cells that we want to look at. And then we add the primary antibodies, which attach to the cells you're looking for. So if we're looking for mesothelial cells, then the primaries would attach to those cells. And then you have the secondaries, which attach to the primaries. And the secondaries are the ones that have the immunofluorescence on it, so you can actually see the color whenever you image them. And then lastly, we just image the slides. So here's one of the pictures from the tissue staining, just to kind of show you what it actually looks like. This in yellow is mesothelin, which is a marker for mesothelial cells. And you can see, as I mentioned, it does form a monolayer and kind of just like encapsulates the cells here. And then the blue is DAPI, which stands for nuclei in cells. So it just kind of shows you where the cells are. And now again, the main thing that we did this summer was work with microfluidics. So to make these, we had to make PDMS molds, and it's just a 3D mold, and we pour PDMS in, and then we punch holes. The holes would be these ports right here, so that way we can actually put the cells in, and again, everything else we're trying to put in. Then we have to autoclave them and bind them to glass, so that way we can actually look at them under a microscope on a glass slide. Then we coat them with poly-D-lysine, which this just allows the collagen to actually adhere to the glass better in the middle, so that we don't have cells in between the glass and the collagen looking like invasion, but it's not actually invasion. And then we fill the center channel again with collagen type 2, and then coat the channels with fibronectin, and the fibronectin just allows the cells to stick to the channels. Then we add the cells, and then for some of the assays, we treated them with TGF-beta, which is just an MMT like signaler. So we would add the TGF-beta 24 hours after the cells if we were going to add it in. Here is one of the assays where we put mesothelial cells in the top channel, which is the green here. And then we put tumor cells in the bottom channel here. So we have mesothelial in the top two, and then tumor cells in the bottom. And this line is OVCAR 8, sorry. And then this top picture is from June 21st. This is when we first seeded the cancer cells. And you can see that there is a little bit of invasion here that kind of went through the post a little bit, but 8 days later on June 29th, you can actually see the invasion towards the tumor cells is greater. This is another assay we did where we actually put the cancer cells in the same channel as the mesothelial cells to see if they would invade through, kind of simulating again that peritoneum and the monolayer where it goes through. So over here, you can see the mesothelial cells with the cancer cells in red here, and you can see that they're going through. And just to show you that they're not just on top of the mesothelial cells, but they actually invaded through, you can see over here on the right, there are the holes where the tumor cells actually like invaded through. Again, this is another similar picture to the one I just showed you. This is in a different device that we did. Again, here are the tumor cells and the mesothelial cells here. And then you can see those holes from where they actually like invaded through and made those holes. And then this one was a picture 72 hours after we seeded the cancer cells. This is using the TGF-beta that I kind of mentioned. So cancer cells actually give off TGF-beta signaling just kind of naturally in the microenvironment, and we think that it induces MMT, which would allow the cancer cells to be able to metastasize better. So here's our control without TGF-beta, and you can see that the mesothelial cells are like more circular, the monolayer is kind of more flush, and they're stuck together. But over here are the mesenchymal cells that have been treated with 10 nanograms per milliliter of TGF-beta. And you can see there's like holes here and they're more like clumped and they're not as like smooth. And that's because again, they're more motile and they want to move around and they don't want to like stick together and just be flush. So our conclusion is that the OVCAR-8 cancer cells invaded through the mesothelial monolayer, that the TGF-beta treatment induced morphological changes. Also, I forgot to mention with the MMT, you could see the structure was different with the cells. Here, I'll go back really quick. The structure was different with these, but to definitively be able to say it induced MMT, we'd have to stain for mesothelial and mesenchymal markers, which would be like one of our next steps. But so far, we can't see morphological changes. So we saw that. And then preliminary data suggests that the mesothelial cells invade towards the cancer cells. And then future directions, just adding cancer-associated mesothelial cells, which have gone through MMT. So taking those cells that have already gone through MMT and then mixing them with cancer cells to compare them to the normal mesothelial cells and see if it makes a difference. And then also staining for mesothelial and mesenchymal markers for MMT, which I mentioned again, just to definitively say, yes, MMT happened. And then also possibly knocking down TGF-beta signaling to slow metastasis. So if it makes it easier to go through the mesenchymal cells, actually stopping the transitioning from happening would make it a little bit harder for the cancer cells to go through. And then just a huge thank you to everyone that supported me this summer, like the entire lab, Dorota especially for spending all the time with me this summer, Dr. Duncan, Serafina, and then everyone at the Hillman site, and some of my references. So any questions?
People on Zoom.
Oh, yeah.
Question. Will you just repeat the question?
Yeah.
And also the Zoom people are welcome to ask a question too.
Yeah.
So is your full strength?
We're really just seeing how they're getting through because the actual logistics of how they get through isn't really studied. So we don't know what to like strengthen about it. We're also just looking at MMT because it does occur. So we were like seeing if there is a difference and if it makes the cancer cells better. So right now we're kind of just studying like the invasion and like seeing how it actually goes through because nobody really knows. Yeah. You mean like, oh yeah. She asked like what substrate I use to mimic the peritoneum. Do you mean like the fluids that we put in? Oh yeah, that was collagen type 2, which is actually really abundant in the body naturally. So that's what we use. The part that mimics the peritoneum is actually that mesothelial monolayer. But again, since the collagen is already abundant in the body, we use that so that way it kind of just is like a middle part if the cells want to move through it or not. Does that make sense? Yeah. Mm-hmm. Yeah. Like almost like the flow of it if they're actually able to get through. So we did have trouble with some, but honestly with the devices we had, it was pretty consistent. So if the flow didn't work, we did have devices where they stuck in the port or they only went halfway through. They're essentially just kind of trash and we didn't use them for actual data. Okay.
Do you
Yeah.
think, as a mechanism that's recruiting the cancer cells, you think it's TGF-beta, maybe the dominant mechanism?
That's like making them be able to metastasize better.
Right.
Yeah, we think that's what we're testing out because it already kind of occurs in the body. So we think that that might actually be one of the reasons why it's able to metastasize. So that's why we're studying it.
Do you think that's the only mechanism? Or are there other
No, I think there's other signaling. I forget what they're called, but we studied other one and there were markers that we studied for other signaling, but I don't think TGF-beta is the only one. Yeah, we just wanted to see how big of an effect it had. Thank you. This up here.